So, coming to the last part of the experiment, DEF16 is actually a major transcription factor regulating hundreds of target genes downstream of the IS. And using the RNA-seq analysis of the N2 control and DEF16 mutants growing on the control E. coli and HNS mutants, from figure 6a, they identified that there is a 319 C. elegans genes that were upregulated by HNS E. coli in a DEF16 dependent manner. It's from figure 6a. And actually, DEF16 targets have been classified in two classes. There is the first class is DEF2 knockout induced class 1 and DEF2 repress class 2 genes. So to their results actually confirm that majority of the targets were distinct from knowing class 1 targets from figure 6a. So they also have done gene ontology analysis, review that their target were enriched in genes associated with body morphologies and multi-cycles, and was depleted for previously identified aging-associated genes. And they suggest that HNS activates the novel DEF16 targets to modulate longevity, and which they classify as class 3 targets. And to confirm the data obtained by RNA-seq, mRNA level would also examine that there is 8 of class 3 DEF16 targets using qPCR from figure 6c. And figure 6d also shows that this target was not upregulated in IIS compromised DEF2 knockout mutants. And figure 6c shows that A class 3 DEF16 targets were upregulated by HNS E. coli in a DEF16 dependent manner. So, coming to the whole scheme of the pathway, that class 1 target, that MG supplements loss in HNS induced upregulation of these 3 class 3 genes. This finding supported the hypothesis that HNS modulates longevity through MG. That is from figure 6e. We could see that the whole scheme is like HNS is acting directly through TORC2 and also down to SGK-1 to inhibit DEF16 and therefore to affect host longevity. That is for the MG effect here. So coming to the end, there is some summary that we need to know that what is the significance of this study. So firstly, low Mg producing E. coli mutants, as E. coli, it adding to extend host longevity in RIP1 SGK1 deficit-dependent manner. And once activated by as E. coli, deficits are regulated novel class 3 target genes. Secondly, it was also found that FUDR, which is the really common use drugs to prevent progeny in C. elegans lifespan assay, it was found that it actually suppressed HNS-induced UPRMT activations. And noting that FUDR did not affect HNS-induced upregulation of class 3 deficit target genes. Next point is Mg is a membrane permeable metabolite that is produced during metabolism in both bacteria and human, and it could induce the formation of AGE, which is implicated in some human pathologies such as diabetes and neurodegenerative disease. And in this study, Mg modulates C. elegans longevity through host signaling, but not through non-specific damages to biomolecules. And using the mammalian cells, they find that Mg enhances the phosphorylation of SGK1 to accelerate cellular synthesis. And the next point is HNS mutants modulate longevity via both Mg RIP1 SGK1 pathway and also CA UPR activity. And we also know that reducing bacteria-derived Mg level might be really used for therapeutic interventions for Mg-associated pathophysiology.